Hello everyone, my name is Martina Mulas and I'm a postdoc researcher at the National Institute of Oceanography and Applied Geophysics in Trieste, in the northeastern parts of Italy. Thanks to the Pyroscience sponsorship to the ICYMARE 2023, this summer I was able to conduct an experiment to investigate the different metabolic rates of Fucus virsoides under different nutrient conditions. Virsoides is an endemic species of the North Atlantic Sea and is the only representative of the genus within the Mediterranean. Once widespread all over the rocky intertidal from the Venice Lagoon to Montenegro, right now is facing a risk of extinction with a high constriction in terms of its distribution. The reason of this decline can be attributed to multiple stressors, such as, for example, the pollution, habitat disturbance, and climate change. Particularly in the Gulf of Trieste, it was recorded a decrease in phosphorus loading from its main freshwater source, with the consequent increase of the NP ratios in the area, which has led towards oligotrophic conditions. Our hypothesis is that the depletion of nutrients can affect metabolic rates as well as photosynthetic efficiency of Fucus virsoides. We therefore query the EMODNet and the Regional Environmental Protection Agency databases in order to extract different NP ratios of the area along the years and then prepare different culture media with artificial seawaters. Apical fronds of around 5 centimeters long were collected from the site and then transferred to the Laboratory of Algology at the University of Trieste. Here they were cleaned from epiphytes and selected by measuring the maximum potential quantum efficiency of photosystem 2. Samples were acclimated to the lab condition for 72 hours and then randomly assigned to different ratios. Meanwhile, we selected 20 milliliters vials provided by the Pyroscience company as incubation chambers. We calibrate the contactless optical sensor at the experimental conditions, and once the setting and the calibration were done, we conducted preliminary trials to define suitable incubation time. The experiment lasted for 2 weeks, and we recorded metabolic rates every 4 days under both light and dark conditions using photorespirometry techniques. Preliminary results show that after 4 days of treatment, there was no difference in terms of net primary productivity and respiration rate. Moreover, we also decided to carry out the same experiment directly in the field, where the species were exposed to a higher irradiance and temperature. In this case, we obtained a much higher results, especially for the net production, which were still in accordance with previously recorded in the area at the end of the 80s. Photorespirometry technique is a very common methodology to quantify the carbon sequestration role of seaweeds. And because this species is at the edge of its extinction, understanding its role in terms of primary productivity is pivotal to stress the importance of its conservation and reinforce the restoration actions. This 4-channel firing O2 instrument, together with all the other accessories offered by the Pyroscience, allow us to achieve our goal and record high sensitive data in a simple way. We want to acknowledge once more the Pyroscience as a sponsor of the ICYMARE and of our experiment, and also to thank the University of Trieste, in particular the Department of Life Science, for offering the facility to carry out the experiment, and the OGS who funded the other expenses.